Hello students, in this video I am going to explain you about behavior of chromosomes during different phases of cell cycle. If we study the behavior of chromosomes during different phases of cell cycle, it will be very easy to solve questions related to this cell cycle, different phases of cell cycle and different phases of mitosis. Okay, let's start with it. So, when a parent cell enters into cell cycle, that is into G1 phase, having two chromosomes. Here two chromosomes are there. Having two chromosomes, then actually in G1 phase, chromosomes are in the form of chromatin fibers. So, the number of chromosomes present in cell during G1 phase is two chromosomes, and these chromosomes are in the form of chromatin fibers. So, how many chromatin fibers are there? Two chromatin fibers. Each chromatin fiber is made up of one DNA molecule. So, the number of DNA molecules present inside the cell during G1 phase is 2. And the amount of DNA, let's say the amount of DNA present in cell during this G1 is 2C. So, as each chromosome or each chromatin fiber is made up of one DNA molecule, it is also called as, the chromosome is also called as monad. This is monad condition of the chromosome or the chromatin fiber. So, this is about the chromosomes during G1 phase. Next, after G1 phase, cell enters into S phase. So, S phase is booked. It is marked for DNA replication or the synthesis of DNA. So, what happens by the end of S phase? DNA replication occurs. So, the amount of DNA gets doubled. So, amount of DNA during G1 is 2C. Now, the amount of DNA by the end of S phase is 4C. But the number of chromosomes remains same. That is 2 chromosomes only. And the number of chromatin fibers gets doubled. So, number of chromatin fibers are 4. And number of DNA molecules are 4. So, by the end of S phase, amount of DNA gets doubled as the DNA undergoes replication, or as the genome undergoes multiplication, synthesis, replication, duplication. Okay, so number of chromosomes are remaining same, but the number of chromatin fibers are getting doubled and the number of DNA molecules are also getting doubled. Let's see why this number of chromosomes remains same though the amount of DNA gets doubled. Okay, actually what we studied in G1 phase, each chromatin fiber is made up of one DNA molecule. So here this is one DNA molecule. This DNA molecule undergoes replication to form its copy, another copy of it. So now this newly formed copy of DNA molecule gets attached with it. So now each chromosome is with two chromatin fibers. Each chromosome is in the form of two chromatin fibers. So that is nothing but 2 DNA molecules. So the number of chromosomes remains same, but the number of chromatin fibers are getting doubled and the number of DNA molecules are getting doubled and the amount of DNA gets doubled anyway as the DNA undergoes replication. So this is the condition of chromosomes by the end of S phase. After S phase, cell enters into G2 phase. So now what is the condition during G2 phase? The same condition will be there during G2 phase as in case of S phase. Next, so by the end of G2 phase, now interphase will be completed. So interphase consists of three phases, G1, S and G2. So interphase is completed. So during interphase, these chromosomes are in the form of chromatin fiber, where each chromatin fiber is made up of one DNA molecule, one double-stranded DNA molecule, which consists of two DNA strands. Right. Now, cell enters into M phase. M phase consists of karyokinesis and cytokinesis. First phase is karyokinesis, which is followed by cytokinesis. Karyokinesis consists of four phases: prophase, metaphase, anaphase and telophase. So, now cell enters into prophase. So, during this prophase, what happens? These chromatin fibers undergoes condensation into chromosomes. So, these two chromosomes having two chromatin fibers each chromosome is with two chromatin fibers, undergoes condensation into chromosomes. Now, each chromosome, so here what is the number of chromosomes during prophase? During prophase, the number of chromosomes is 2 again. Number of chromatids here, number of chromatids, not number of chromatin fibers. Number of chromatids is 4. So what can we say? One chromatin fiber gets condensed into one chromatid. One chromatin fiber gets condensed into one chromatin. So two chromatin fibers of a chromosome gets condensed into two chromatids of a chromosome. Now number of DNA molecules. How many DNA molecules are there? 4 DNA molecules, because one chromatid or one chromatin fiber is made up of one DNA molecule. So number of DNA molecules are again 4. Amount of DNA is again 4C. Amount of DNA is again 4C. Next, after prophase, cell enters into metaphase. So in this metaphase, maximum chromosomes exhibit maximum condensation. So this is the best phase to study the number of chromosomes and even the morphology of chromosomes. So by the end of metaphase, these chromosomes align at the center of the cell to form equatorial plate or metaphase plate. That movement of chromosomes towards the center of the cell is called as congregation, and alignment of chromosomes at the center of the cell is called as equatorial plate or metaphasic plate. Right. Now this the number of chromosomes, number of chromatids, number of DNA molecules and amount of DNA is same in metaphase as in case of prophase. Next, coming to anaphase. So after metaphase, cell enters into anaphase. So what happens during anaphase? Centromere splits. After splitting of centromere, chromatids of a chromosome start separating from one another. So these are the two chromatids of a chromosome and these are the two chromatids of another chromosome. They separate from one another and they reach towards two opposite poles of the cell. So what can we say here? The number of chromosomes. How many chromosomes are there here? Number of chromosomes are 4. Number of chromosomes are 4 in case of anaphase. This is in case of metaphase. And even in case of prophase, same thing. So now, number of chromatids. Chromatids are again 4. So here, what can we say? Here, one question we can discuss about one question. So, in which phase, during which phase of mitosis, the number of chromosomes and the number of chromatids will be similar. So, the number of chromosomes are 4 and even the number of chromatids are also 4. Okay. In which phase of mitosis, the number of chromatids are similar and the number of chromosomes are double the number of chromosomes present in prophase and metaphase, that is anaphase. I just repeat this question. In which phase of mitosis the number of chromatids are same but the number of chromosomes are double the number of chromosomes present in prophase and metaphase? So in what phase? Anaphase. In anaphase only the number of chromatids are same. Here number of chromatids are same in anaphase. In prophase and metaphase also number of chromatids are same, 4 only. Here coming to number of chromosomes. In anaphase number of chromosomes are 4. 1, 2, 3, 4. Number of chromosomes are 4. But in case of metaphase and prophase number of chromosomes are 2. So during which phase of mitosis the number of chromosomes are double but the number of chromatids are similar with respect to prophase and metaphase. So that is anaphase. Answer is anaphase. So by the end of anaphase, these chromatids, which are the future daughter chromosomes, which become the part of daughter nucleus, reach towards two opposite poles of the cell. After reaching, then cell enters into telophase. So during telophase, what happens? These chromosomes or chromatids undergo decondensation. That means they undergo decondensation back into chromatin fibers. That means they are losing their identity. They are not visible like chromosomes. They will be in the form of chromatin fibers. So here, after decondensation, what happens? So this is the condition of the cell. So at one pole, like this, chromatin fibers, 4 chromatin fibers, and on the opposite pole, 4 chromatin fibers. Like this, nuclear membrane will be reformed. So two daughter nuclei will be formed by the end of telophase. So in telophase, how many chromosomes will be there? 4 chromosomes, which are in the form of chromatin fibers. 2 chromosomes towards one pole and 2 chromosomes towards the opposite pole. So this pole becomes one daughter cell after cytokinesis. This pole becomes another daughter cell after cytokinesis. So after cytokinesis, now let's see the configuration of these chromosomes, chromatids or chromatin fibers, DNA molecules and amount of DNA present in each daughter cell. Now after cytokinesis, in each daughter cell, how many chromosomes are there? Number of chromosomes present in each daughter cell is 2, because at one pole 2 chromatin fibers and on the opposite pole 2 chromatin fibers among 4 chromatin fibers. So number of chromosomes is 2, number of chromatin fibers is also 2, number of chromatin fibers is also 2. Next, number of DNA molecules is also 2. Now amount of DNA is 2C. Because half of the amount of DNA is distributed to one daughter cell and remaining half is distributed to another daughter cell. Among 4C, half is distributed to one daughter cell and remaining half is distributed to another daughter cell. So amount of DNA present in each daughter cell is 2C. Okay, now let's observe this configuration with respect to parent cell which enters into G1. So, what is the configuration of this chromosomes, chromatin fibers, DNA molecules and amount of DNA of the parent cell when it has entered into G1 initially? So, this is about daughter cell. Number of chromosomes 2. There also number of chromosomes. In parent cell also number of chromosomes are 2. Number of chromatin fibers in daughter cell are 2 and even in parent cell which has entered into G1 is also 2. DNA molecules also 2 and amount of DNA 2C. The same configuration in the parent cell. So hence it is called as equational division. So this is all about the behavior of chromosomes during cell cycle. So in the next class I'll give you some tricks to solve questions related to this. Numerical based questions or some problem based questions related to this cell cycle, different phases of cell cycle and different phases of mitosis. Thank you students.